everyone, and welcome to today's live broadcast, Developing Bioinformatics Approaches to Understand the Functional Consequences of Gene Expression Variability, presented by Dr. Jessica Marr, Assistant Professor at the Albert Einstein College of Medicine in the Department of Systems and Computational Biology in the Bronx, New York. I am Marjorie Torres of Labroots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Labroots. Labroots is a leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Please join me in welcoming Dr. Jessica Marr. I will now turn the presentation over to her.
Hi, everybody. Well, thank you so much, Marjorie, for the kind introduction, and many thanks to Labroots for the opportunity to present some of the research that we've been doing in my lab. I'd like to echo Marjorie's comment about questions. I'm more than happy to take questions that you may have after viewing this presentation, so please feel free to send them through. I'm going to go ahead and get started and talk about the focus of the work in my lab, which is centered around the idea of gene expression variability. So as Marjorie mentioned, I am part of the systems biology department at Albert Einstein College of Medicine. And one of the big things that we do as systems biologists is we really want to understand what gene regulatory networks look like. And so this stems from the phenomenon where we now live in a world that we know that cells operate through the integrated action of many different genes. We understand that one gene, one phenotype is no longer a paradigm that holds. And so as systems biologists, what we spend a lot of time thinking about is how to model these networks accurately. So for instance, what genes go into these networks, and also how does information flow through these networks to control and regulate different cell phenotypes. So when we think about how networks operate, we understand that certain genes that are in these networks are controlled by different levels of signal. But what we're coming to recognize more recently is that noise plays a big feature in regulating these networks too. And in the recent past, starting, more in some of the papers in 2010 and 2011, there's been some beautiful work that's been done in the context of C. elegans. And I've listed one example here in Raj et al., who showed that actually there was an increase in gene expression variability that was downstream of a mutated gene. And this variability that was seen at the gene expression level was in fact explanatory of the incomplete penetrance associated with the mutation in this particular gene. Berger et al. In 2011 had another Nature paper that showed actually the link in synthetic lethality and the expression variability of one of the synthetic lethal interaction partners. And he showed that also in C. elegans that increases in gene expression variability of the synthetic lethal partner actually determined, again, the incomplete penetrance of the trait. So we have these examples that come to us from model systems that demonstrate how important the variability of gene expression is in the context of these regulatory networks. And so when we think more generically about the signal transduction pathways that we know are relevant to every single cell, there is this importance of understanding how signal and noise contribute to the proper regulation of these networks. And so you can think about a generic signal transduction pathway that's made up of different molecules that have different roles. So for instance, receptors, kinases, or transcription factors. And if we think about surveying or capturing the transcriptional snapshot of this pathway from cell to cell or from patient to patient, we can imagine following the level of variability or consistency in these gene expression values. So you can imagine that the distribution of expression variability is not equal throughout this pathway, and paying attention to those extremes or degrees of variability can actually tell us about transcriptional stability or the degree of regulatory constraint. So if you think about this a little bit further, the idea that some genes may have low variance is indicative then of tighter regulatory constraints or control in the system. And on the other side, we can think about high variability or high variance, reflecting greater heterogeneity, which may speak to increased functional plasticity or a greater diversity of cell fates that might be the outcome of those particular genes. So I'd like to emphasize that variability is really an inevitable component of any biological system. And as our tools and technology improves, we're really being reminded of this fact with more and more data that we collect. But when we think about why this is the case, why this is such an inherent feature, it's pretty clear from the gene expression perspective. And that's because we know that gene expression is the product of a series of biochemical reactions. So as we know, for a transcript to be made, there needs to be a whole set of different molecules that have to come together physically in order for this process to move forward. And some of these components, like transcription factors or cofactors, can either be spatially regulated and also present at low concentrations, so that the actual event of producing a transcript is a probabilistic event. For housekeeping genes, that probability may occur with 0.99999% or 9 high certainty, but there's still that small element of stochasticity there. And that's the part that I'd really like to emphasize here, that when we look at the distribution across the transcriptome, there is stochasticity there in the gene expression of certain genes. And what we're coming to understand is that some genes have variability that seems to be buffered or compensated for by other processes and have no effect on the phenotype. And in other circumstances, there is variability in some genes that can lead to quite distinct phenotypic outcomes. So one of the focus points of my lab is really to understand what is it about certain genes that seem to be buffered and others that have variability that's really quite key in the phenotypic consequences of that distribution. So another way to think about gene expression variability is in the context of looking at distinguishing factors when we're trying to understand regulation of a particular phenotype. And in gene expression, what we often do is we use a statistic like a t-statistic to understand the genes that are differentially expressed between one phenotype or another. So you may recall that the t-statistic is just a ratio of the average expression difference between two groups that's divided by the amount of noise or variability in the two systems. And we can see this graphically. So for instance, if we're comparing a disease group and a control group, then the genes that we are going to pick up as differentially expressed are those that have the biggest difference in average divided by the most consistent expression. So those genes that are very consistent will have a smaller degree of noise, and therefore the overall t-statistic that we find will be much larger and therefore more likely to be significant. And this is a very sensible way of identifying genes that are important to one phenotype or another. But one of the limitations is it misses out on changes in gene expression variability. And I've got two cartoons here to illustrate what I mean by that. So for instance, there's one example where, for instance, the variability of the gene in the disease group may be much more heterogeneous, whereas in the control group, the stability of that gene is quite tight or quite small. And the more extreme example on the right side is where on average this gene is expressed equally in the control and the disease group, but you can see there that the variability is quite different. So in the disease group, the expression of this gene is much more heterogeneous. That may be due to an upstream mutation or SNP in the promoter region or some other regulatory factor associated with the disease. And if we were only focusing on the T statistic, we would miss this detail. So I think there is a reason to look at both changes in variability as well as changes in expression to really understand the underlying regulators of a specific phenotype. And so some of the earlier work that I've been involved in has been looking at this specific question in the context first of human stem cells that we collected from neurological diseases, for instance, Parkinson's disease and schizophrenia. And so using a stem cell model that was collected from olfactory cells and turned into neural cells through these olfactory neurosphere-derived cells, what we did was we were able to look at patient variability in these stem cell models of these disorders, which, as you can appreciate, are very difficult to study in living patients. And so the study we did was published a while ago in 2011 in PLOS Genetics. And basically what we did was we looked at changes in gene expression variability between these different patient groups as well as a group of related controls. And when we looked at specific pathways that were relevant to these stem cell populations, we noticed that there were differences in gene expression variability such that there were increases in the Parkinson's disease group and decreases in the schizophrenia group, and for the controls there was somewhere in the middle. So this really suggested that variability at either extreme was present in a disease state. And we looked further at the functional roles associated with these changes in variability. We noticed interesting things in terms of differences in cell surface receptors as well as transcription factors, and also the connectivity of these genes in terms of the different protein-protein interactions that they operated in. So more recently, what my lab has been thinking about is how we can use variability, but also some other related statistics to really understand the regulatory distribution of genes in a particular population. And so what I want to do is take you back to perhaps high school statistics, where we often look at the probability density of a specific data set. And so in this example, what I have is a density that's been drawn for a single gene that's been captured in a single cell population. So each point along this density represents the gene expression level in a single cell, and we have a population of cells here. So statistics tells us different aspects about this particular distribution. And you're all familiar with, I'm sure, the first moment, which is the median or the mean. It measures the center of this distribution. And when we think about these moments, these are these are statistics that report on different aspects of the population in the data. So our first moment is the median and mean. The second moment is the variance, which measures the spread of this distribution. But if we keep going, there are actually higher moments above the second moment that we don't often always talk about. And so the third moment is skewness, which is something you may have already heard of. And as we keep going, we have hyperskewness, hyperflatness. And as we move further, the interpretability of these moments tends to get a bit vague. But what is really interesting is that there are biological features that we can extract from these higher moments and extend them too to look at other features such as bimodality or multimodality, which is certainly a feature that's relevant for single cell data. So what I hope to do is talk to you about some of the ways that we're using these moments to infer more about the underlying biology. And I think this is really relevant and important because in bioinformatics, often our methods really only focus on that first moment, and there really is a lot of biology to be explored if we go higher. So what I want to do today in this presentation is talk about 3 separate projects that we have in the lab. One of them starts off with dealing with a method that we've developed to understand gene expression variability in the context of pathways. I want to then talk about cancer biology in 2 areas where, one, we've been looking at changes in gene expression variability to look at genes that are much more tightly regulated in the tumor versus normal. And finally, some examples of how we've used skewness in gene expression as a way to understand regulation of tumors in a different way. So without further ado, I'd like to start off by introducing pathVar. And this is a tool that we developed in my lab. It was recently accepted for publication in PhD, and it's been available in Bioconductor as a standalone package for over a year now. And this is work that has been done by my postdoc, Laurence Tetrante, and my programmer, Sam Zimmerman. And it really centers on this very basic idea of thinking about the distribution of gene expression from patient to patient, or if we're looking at single cells, from cell to cell, and understanding what is that distribution of variability across the pathway that we have in our database, or the gene set that we've collected from a particular study. And we can actually use that information in the form of the distribution of variability that we see in panel B, where some genes in this pathway are expressed with much lower variability versus other genes that are much more variable. And we want to use this distribution to understand which genes belong to either of these extremes, to understand how transcriptional stability or regulatory constraint is distributed within this particular pathway. So the pathVar method can function in a 1-group setting or a 2-group setting, and I'll give you some examples of what I mean by that. But essentially, the outline of this method is quite straightforward. Step 1 starts with a diagnostic plot that helps the user determine which statistic to use to measure variability. The second step is to then use the statistic to analyze for each pathway the levels of gene expression variability that we see, and assess whether this distribution of low, medium, high variable genes is different or aberrant, either from the reference, if we're working in a 1-group setting, or between the 2 phenotypes that we're contrasting. And then there's a few different visualization steps to facilitate proper interpretation. So in terms of measuring variability, this is something that there are several different statistical estimators that can be used for this purpose. And as with all statistics, there are pros and cons with using one versus another. In terms of looking at expression data, what we found is that a good way to decide on which statistic to use can be determined by looking at the correlation or the distribution between the variability statistic and average expression. And the rationale behind that is that what we see sometimes is that there is correlation between these 2 statistics. And what we want to do is select a variability statistic where there's less correlation or least correlation, because that way we can investigate changes in variability that aren't purely recapitulating the changes that we see in average expression. So we did some work into looking at which of these estimators performs better than the other. This is work that's available in a PLOS Genetics paper that we had in 2015. We did various simulation studies, and we found that the standard deviation often is a more reliable or less correlated estimator, but I want to highlight that this is not something that happens in every single data set, and in fact it seems to be data specific, and this is something that we're still working on and hoping to add more clarity to, but this is really a reason for investigating how your estimator behaves in your data, because it can vary depending on the data that you're looking at. So if we go back to what pathVar is doing, if we take this example of comparing the distribution of low, medium, high variability genes in a pathway versus a reference, like this versus this, there's two comparisons that are occurring here that we care about. So the first comparison is this here, where basically what we're trying to do is test the null hypothesis that the reference and pathway share the same variability count distribution. Okay, so basically, does the histogram on the right look the same as the histogram on the left? And the way we assess that statistically is through the use of a chi-squared test, which has asymptotic assumptions, which means it becomes more accurate as the sample sizes increase. Or we can also use a multinomial statistic, which is really nice because it's an exact statistic, so it computes the p-value exactly. The second kind of comparison we're also then interested in testing is between the same level of variability. So for instance, this one here, for the low variability counts, we want to compare whether this number in the reference is the same as the number here in the pathway 1 that we're testing. So this is only looking at low to low between the two groups, and this is done through a binomial statistic, where now assessing whether this specific pathway has a different number of low variability genes than we'd expect compared to the reference. So what I wanted to do next then is show you an example of what I mean. And here what we've done is we've used data from the Bock paper, which appeared in Cell in 2011. This was microarray data that profiled two populations of cells. There were human ESCs, embryonic stem cells, and they wanted to compare the gene expression profiles observed with induced pluripotent stem cells. So this data is a little bit old, but we think it's relevant just to highlight differences that we're seeing here. And so the three pathways that were significant, among some others that were quite interesting that we wanted to highlight here, were KEGG pathways. So these were the spliceosome, oxidative phosphorylation, and ECM receptor interactions. And you can see that the shapes of variability distribution are quite different from the reference. And in this case, the reference is all the genes that were profiled on the microarray with some initial filtering steps for quality. So what we've done with this pathway is, what we've done with this package and this method is we've tested it extensively in different kinds of data platforms. In addition to microarrays, we've also tested this with RNA-seq data. We've tested this on single-cell data sets as well as bulk, human, mouse, host parasites, other stem cell populations, and that's all part of the paper that you can find in PHA. So in the two-group comparison, instead of comparing ESCs to the reference, we can also compare ESCs directly to the changes that we're seeing in induced pluripotent stem cells. And so this is another feature that may be more relevant to the question that you're interested in exploring with your data sets. And the same kind of principles apply. So here, what we've done is highlighted two KEGG pathways that were differentially variable between the two stem cell populations, so oxidative phosphorylation and DNA replication. And you can see that there's quite different structures here. So for OXPHOS, for instance, we see a much higher representation of low variability genes in the ESC population. And for instance, in the DNA replication side, a subset of those genes seem to be much more variably expressed in the iPSC population, as you can see from this bar here compared to the height of this bar here. So you can see that the interpretation is quite straightforward. And then through the software, what you can do is you can then pull out the genes that are present for these particular highly variable genes and understand which parts of the DNA replication pathway or gene set are much more variably expressed in iPSCs versus ESCs. So this package, as I mentioned, is available on Bioconductor. Please feel free to check it out, and if you have any questions or concerns, please let me know, and we'd be happy to understand how we can make the software more relevant. So what I wanted to mention too was also the extensive testing that we've done, but also in the context of other methods Obviously, in bioinformatics, there's lots of different methods out there for understanding how pathways contribute to changes in transcriptional regulation. And so what we wanted to highlight was how pathVar was either different or overlapping with other methods that we commonly use. And so what we've done is we've used GSEA-LM, which is an R package from the Gentleman Lab that was used to look at gene set enrichment through a linear model. We've also used gene set enrichment in a different context where we're contrasting the distribution of p-values. In this case, it's using lemma to assess differences between the two groups. And so here in this more traditional version of GSEA, we're looking at changes in those p-value distributions. And so here I've only got the counts of the pathways. If you're interested in understanding what those pathways are, I can definitely share those with you. But essentially what we see is that there are some pathways that are uniquely picked up by pathVar, our method, and others that are uniquely picked up by the GSEA approaches. And then there's ones that are also in common. We also looked at using pathVar where instead of based on the variability, we also base this around average expression. And so this was a way to highlight how our method structurally might compare if we were looking, instead of variability, we were looking at differences in low, medium, and high average levels of expression. And so there we see overlap, considerable overlap depending on the comparison that we looked at. And just to highlight in a bit more detail, we looked at a few different comparisons, 3 from different Cancer Genome Atlas data sets, the BOC data set we used, as well as a stem cell data set looking at Down syndrome versus wild type or control cells. We also looked at a non-human data set from a neurological study done in mice. So what I'm going to do now is just switch gears and hopefully stay within the time frame. But I wanted to talk about another study that one of my students, Cassandra Litchfield, did more recently, where she was really interested in looking at the distribution of gene expression variability across different tumor types. And so the kind of question that she wanted to answer was whether we could find new regulators based on changes in variability. And specifically, did these regulators, were these different genes or the same across different tumor types? And the kind of rationale or motivation for this study started with a paper that we found from 2008 in PLOS Genetics, where they looked at decreases in gene expression variability in tumor versus matched control samples. And this was a very illuminating study. They found that genes that had much tighter expression in the tumor versus matched control had quite a functional effect on the viability of the tumor. And so the study was done a little while ago, and it was done in the context of microarrays. And what we have the opportunity to do now is to really look at a modern version of the study in the context of the Cancer Genome Atlas data, but also with RNA-seq data that has many, many more samples than this original study. So what Cassie did was she took 12 different tumor types, and these all had matched control samples. You can see the representation of different sample sizes that are in the table there. And what she did was she used the CV, which is defined as the standard deviation divided by the mean, to identify a subset of genes that were tightly regulated in cancer. And mathematically, how she found those was to define those genes that fell in the bottom 15th percentile of the CV in the tumor and above the 15th percentile in the CV of the normal samples. So basically, these genes correspond to the yellow dots that are being shown by that blue arrow, and now also this red arrow too, because what they're falling then in the lower set of this sorry, I'm just confused by the arrow here. Okay, by the distribution here in the CV, and much higher in the normal. So for whatever reason, these genes have a much tighter expression distribution in the tumor samples. And so she identified this subset of genes in each of the tumor types that she was looking at. And then she wanted to answer the question or investigate the question of whether there was overlap or similar groupings in the tightly regulated genes that she'd identified from tumor to tumor. And so what she did was she did a hierarchical clustering here to cluster based on the overlap. And we saw some interesting things where some tumor types clustered together. So for instance, you can see that the lung cancers clustered together here. Also thyroid and prostate cancers also cluster together. We think this is because of the hormonal aspect that these two tumors share. You'll notice that these kidney cancers also cluster together, but in fact, this kidney cancer does not. And we think that that could be due to a difference in aggressiveness of these tumors. But there are more things we need to look into for this study. So what is also interesting is that the structure of this dendrogram recapitulates what was also observed in the context of average gene expression. And this was a study that was done more recently by Peng et al. In 2015. And there's a really fantastic paper there in Scientific Reports that looks at these different tumors as well, but from the context of average expression and differential expression. So what we wanted to do then is, given that there did seem to be overlap, was look for a common subset of genes that were tightly regulated to a core of these tumor types. And so when you look at how many genes are retained when you increase the overlap, we saw that for 9 out of 12 of the tumors that we were considering, there are 9 genes that are common to all 9 tumor types. And this was starting from an original set of about 18,000 genes. So these are the 9 genes that are tightly regulated in all 9 of the 12 tumor types. You may recognize some of the genes that are in there. And what was quite interesting, we thought, was that when you keep increasing that threshold from 9 up to all the way at 12, you do actually, when you consider 10 cancers, see a subset of these genes that are still in common. And what's interesting about some of these genes is that they're known to have prior roles in cancer. So these genes were selected purely based on their tight regulation, so a purely data-driven selection. And so it's somewhat interesting that they have roles that we already know in terms of their influence on tumor regulation. So another thing that Cassie did was to apply pathVar to investigate the changes that we saw in different pathways with respect to expression variability by comparing the matched controls to the tumors. And so what she saw was really quite interesting in that a subset of pathways were significant that were quite relevant to cancer biology. And she used KEGG pathways in this instance and used significance as defined by less than 0.01 p-values that were adjusted for multiple testing. And so we've used this example from p53 signaling pathway. As you probably know, this is a very important and relevant pathway to cancer biology. And you can see based on the distributions that the matched controls actually have many more tightly regulated low variability genes than the tumor here. And so when we look at the other pathways that Cassie found, these were things like DNA replication, ubiquitin-mediated proteolysis, and mismatch repair. We actually see the same trend where there's a loss or an increase in heterogeneity in the pathway. So we see, for instance, the much more tightly regulated genes here that drops off in the tumor. So we think this speaks to the general instability or destabilization of key pathways that occur in the tumor, which obviously we know about through other studies or through other research. And I think the next steps here are really to understand how this pathway is contributing. Obviously, there's parts of the pathway that are being destabilized, but there's also potentially parts of the tumor that are being more tightly regulated, which is some of the earlier stuff that I presented. So putting both of these pieces together gives us more comprehensive understanding of how transcriptional regulation is being perturbed or altered in the context of these tumors. So finally, what I wanted to talk about was stepping away from expression variability, but moving on to the next moment, which you might recall is the skewness statistic that we talked about. So this was a project that was pioneered by one of the students in my lab, Ben Church, and with more recent assistance from Henry Williams. And both Ben and Henry were really fascinated by this idea of how skewness could be used as a way to both model and then understand gene expression heterogeneity. And so we don't really tend to talk about skewness in a quantitative setting, but qualitatively we understand this to be instances where distributions show a lot of asymmetry. So this asymmetry can be positive or negative by both of these densities that we've got. And in terms of quantifying the skewness, Ben derived a statistic that takes into account both the skewness, the third moment that we can calculate from a distribution, but this is standardized by the standard deviation, the second moment. And just wanted to point out that by a theoretical estimation, this is a biased estimator, but the actual degree of bias, especially for the size of the data sets that we're working with, is really quite minimal. So this isn't a huge concern. But just to give a bit more background about why, instead of just taking the third moment, we've normalized this statistic by the standard deviation as a way to really highlight the asymmetry that we're seeing in the data. And so just using the skewness statistic, we're not necessarily able to distinguish distributions like in the right-hand panel that clearly are showing deviations that we would like to model in the gene expression distribution. So what Ben and Henry did was they developed a pipeline to investigate skewness using different data sets, including the Cancer Genome Atlas. So what they did was they applied the skewness distribution to different gene expression data sets in a normal and a cancer setting. They also developed a statistic to determine the stability or the convergence of this particular skewness estimator so that we could accept or reject data sets that were insufficiently large. So we accepted those data sets that showed reasonable convergence, so we were fairly sure that the accuracy, at least, of this data was, or the accuracy of the skewness statistic was acceptable. And so what he did was then he looked at genes that changed dramatically their skewness in the cancer versus normal. So maybe the gene was extremely skewed in the cancer group, but in the normal data, it was symmetric. And when we look at what those genes represent, they largely represent different metabolic or translational pathways in the RNA-seq and the microarray data. And Ben and Henry found some slight differences in the microarray versus RNA-seq data, but in terms of understanding, we are interested more, I think, in the RNA-seq data that we're seeing. So another thing that they did was then to try and tie the skewed expression that they saw with changes at the level of DNA methylation. So one of the nice things about Cancer Genome Atlas data is that we have access to other data types that have been profiled on the same patient. So what they did was they looked at genes that were significant for changes in methylation, and also looked at how skewed they were in these cancer populations. So they tried two different approaches to determine which was the most significant subset. Basically, the two approaches were fairly similar, and from this, they basically ranked the top, the set that were most skewed with the differentially methylated loci, and they took the top 500. And from that, what they wanted to do was look at the relationship between DNA methylation and the skewed genes in terms of expression. And so what you see here are different relationships here for the kidney renal cell carcinoma cohort. And these are all, the different points are colored based on the quadrants that they fall into. As you may or may not know, the way we interpret DNA methylation really depends on where those loci are falling. So the context really matters or is helpful to know. So we divided this data up into where these loci were annotated to, for instance, promoter regions, gene body regions, and the UTR. So what they found was that there is a very negative relationship or association between genes based on their skewness and also the change in N value, in other words, the change in DNA methylation. And they used confidence intervals to assess the significance of these Pearson correlation coefficients. And you can see that for each of the 3 different genomic regions that we looked at, there's quite a significant negative correlation there. So this is quite interesting. What they wanted to do then was to determine, given that this is based off the top 500 genes, whether that subset selection had any influence on the negative correlation that we're seeing between skewness and DNA methylation. So what Ben and Henry did was they downsampled, going from 500 all the way down to 100 of the significant genes, and they did this multiple times to ensure that they had a generalizable result or a stable result. And you can see in this graph that they did this for the 3 different regions that we were interested in, the UTR, the promoter, and the gene body region. And you can see that basically the negative correlation as you go from the top 500 to the top 100 genes actually gets stronger. So it gets more negative, which is reassuring because it demonstrates that this correlation we're seeing is robust to the size of the significant genes that are being considered. Okay, out of all the different 3 different marks, we see that the promoter probes, the loci that were located in the promoter regions, have the greatest robustness compared to the other regions, and it also has the most negative correlation. And this suggests the link between the skewness and the DNA methylation we're observing. And this makes a lot of sense given what we already know about methylation of these loci in the promoter regions and its relationship to gene expression. The negative correlation we're seeing in the gene body is also quite interesting, and we are still investigating that further to really understand what that means. What I'd like to do then is end the presentation by acknowledging the people in my lab and all our collaborators that have worked really hard on these different studies. The pathVar work was done primarily by Laurence de Tarante and also by Sam Zimmerman. The cancer tightly regulated study was done by Cassie Litchfield, who worked with me over the summer. She's based at the University of Melbourne. And finally, in looking at skewness, this was done by extraordinary work by Ben Church and Henry Williams. And we are really fortunate to work with a whole host of different collaborators, both at Einstein and located elsewhere. And I'd like to thank you for tuning into my presentation. Thank you.
Thank you, Dr. Marr, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your question into the box that appears on your screen and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, what are some possible interpretations for decreased or increased changes in gene expression variability? How could we use the other resources to further investigate the underlying mechanisms or functions that drive variability in gene expression?
So that's a really interesting question. It's very broad. And I think there's many different ways to answer that. But I think in thinking about increases or decreases in variability of expression, this could be, for instance, due to, in the case of increased variability, could be due to mixed cell populations. So, for instance, these cells may just have very different transcriptomes because they have different cell fates or different cell types. In the case of increased or decreased, gene expression variability, this could be an indication of tighter regulatory control. So for instance, housekeeping genes or critical transcription factors or critical cell surface receptors. So I think there's a very functional interpretation to what these changes in gene expression variability can lead us to. The ways of using further resources to understand what those functions could be or what the mechanistic interpretation of variability could be, there are lots of ways that we can approach that. Informatically, we could use other annotation resources. So for instance, like the gene ontology that has different ontologies like molecular function or cellular component or biological process. We could look at molecular function to try to understand if certain cell surface receptors are more variable or less variable. These are sorts of things that we could integrate in the context of this sort of analysis.
What are some of the possible limitations to using interpatient variability as a means to identify tightly regulated genes? Are there other kinds of experimental data that could be used to supplement or augment these interference?
Yes, so one of the really great things about working with patient cohort data is that we have a means to really understand what are the disease profiles like in terms of modeling those changes. But one of the limitations is that patients can have different transcriptomes or different changes due to a variety of other factors. And they may not necessarily be relevant to the disease or the genetics of the disease that we're really trying to get at. So there are issues with modeling inter-patient variability because these patients might be on different drug regimes. They may have differences, in terms of there may be gender-specific differences that are influencing the changes that we're seeing. They may be in different environments, all sorts of factors that we haven't controlled for in our analysis. And even if we did, given the way the data was collected, we may not have the design set up that we can actually account for the influence of each of these different factors. So there certainly are some limitations when working with patient data and using inter-patient variability as a surrogate for understanding regulation of a specific pathway. But there are some advantages too, so it's good just to keep both of those in mind. In terms of addressing those limitations and thinking about what other experimental data or platforms out there may be useful, I think one area that's really exciting right now is single-cell profiling. So for instance, single-cell RNA-seq or some of the fluidine methods or 10x. These are all really great platforms that allow us to look at what is the distribution of gene expression in terms of cell to cell across a whole population. And I think this is really fascinating because it means that we can look at a population of isogenic cells. So all the genetic background of these cells is all identical. Therefore, the changes that we're seeing at the level of gene expression are due to basically differences in the system. So we can really start to understand the stochasticity that's occurring across the transcriptome and start to understand the functional consequences. So I think integrating or augmenting the things that we find in the context of patient populations with what's actually going on then at the single cell level would be really, really fascinating and powerful.
Integrating gene expression and DNA methylation in the context of skewness highlighted distinct correlative structures. What other data types could be integrated with gene expression heterogeneity to further understand transcriptional regulation?
So I think in this day and age, there are so many different kinds of data sets that we can integrate. And it used to be a bottleneck of our technology that we couldn't necessarily do these kinds of coupled experiments where we could grab the RNA and then the DNA or the methylation profiles of the same sample. But nowadays, a lot more of these coupled methods are becoming accessible and reliable. And so I think it's actually more of a bottleneck of our bioinformatics models that need to step up and be able to integrate this data. I think for gene expression variability, methylation for us was a very obvious first step to integrating the changes that we saw at the level of gene expression with what was going on in methylation. But there are other data types that would be, I think, equally illuminating of how the system is being regulated. One of those is to look at genetic variation. So an obvious question would be upstream in the promoter region of these patients, can we find a subset of SNPs that may be relevant to one subset of patients versus another that would lead to increased or decreased variability in the expression of that transcript. Another type of data set that would be really interesting to look at would also be at the proteomic level. So the changes in gene expression variability lead to more stable abundance of certain proteins, in which case there would be a very interesting functional interpretation. And I think proteomics is an interesting area because in terms of protein stability and variability, there are certain distributions that are being modeled nowadays, and we know about that. So depending on half-lives and different distributions like the gamma or the beta. And so correlating that with what we see in terms of stability of gene expression across the population would be quite interesting too.
I would like to once again thank Dr. Jessica Marr for her presentation. I would also like to thank Labroots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August 10, 2017. You will receive an email from Labroots letting you know when this webcast will be available for replay. Please share the announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again. Goodbye.